Okay, so the four types of enzyme inhibition. This is also another very high yield topic on the MCAT. So competitive inhibition is the easiest, most intuitive type, I think. So in this type of inhibition, the substrate competes with the inhibitor to access the active site. So the inhibitor usually resembles the substrate, but it does not elicit a response or form a product. So that's exactly what it is. It's competitive because the inhibitor is actually competing with the substrate, and that is the mechanism by which it inhibits. It's competing, it's outcompeting the substrate. So a good competitive inhibitor would have a higher affinity for a substrate than, would have a higher affinity for the active site than the actual substrate. And this type of inhibition, though, can be overridden by increasing the substrate concentration to a much larger value than that of the inhibitor. Because think about it. If we're talking about mass action kinetics at that level, if you add more of the substrate concentration, it's going to be, if you increase the substrate concentration, it's going to increase the likelihood that the substrate is what reaches the active site rather than the inhibitor. So by adding more substrate, we can actually override the inhibition. So because of that, Vmax is not affected. We can still reach that same maximum rate if we add enough substrate. However, we're going to need more substrate to do that. So the Km increases, which means that apparent affinity is going to decrease because we need more to elicit the same response. So the affinity is going to decrease, Km would increase. And right at the bottom, that's the Michaelis-Menten and the Lineweaver-Burke plot. So you can look at the Michaelis-Menten, it levels off right over here at Vmax. Vmax is not affected in both of them, but Km changes, as you can see. And the same thing over here. If we look at the Lineweaver-Burke plot, Vmax is not changing. It's the same point right over here for both of them, but that Km value is going to change. So this is what it looks like on a Lineweaver-Burke plot. And then we can use those variables. We know those variables to actually find out the values using that Lineweaver-Burke plot. So the second type, we have non-competitive inhibition. So these types of inhibitors bind at an allosteric site, not the active site, like in competitive inhibition. So we're no longer competing for that active site. They're going to a completely different location than the substrate is. So since the inhibitor is not competing with the substrate, you can't override it by adding substrate. So if we have an enzyme that's inhibited allosterically, it doesn't matter how much substrate you add, that enzyme is inactivated. It's not going to work. So because we can't increase activity by adding substrates, the Vmax is going to be lowered and the Km is going to stay the same because the substrates are still binding, remember, they're just inactive. So binding and affinity remains exactly the same, but the Vmax is going to be lowered. And you can look to the right, I put the Michaelis-Menten plot up top. We can see that Km value remains exactly the same, but the Vmax decreases. So this one is the uninhibited reaction, and this two is the inhibited reaction. And you can look right over here, no inhibitor present and inhibitor present, you can see right over here the Vmax does change. If you look at that that y-axis, the Vmax changes. However, the Km value does not change. It's that same point right over there. So third, we have uncompetitive inhibition, and it's important to distinguish between non-competitive and uncompetitive. I don't really have a great mnemonic to distinguish, but I encourage you to find up make up your own or find one that helps you, because it can be kind of confusing. And they are similar, they do work in similar ways, but it's very important to know that they're different. So during this type of inhibition, the inhibitor will bind only to the substrate enzyme complex. So non-competitive means we're binding to the enzyme before it forms the substrate enzyme complex. Uncompetitive means we can only bind after the substrate enzyme complex is formed. So these, like non-competitive inhibitors, bind to allosteric sites. So again, we're not competing for the active site, but uncompetitive inhibitors only bind to the substrate enzyme complex. This also decreases the Vmax of the reaction since we can no longer reach the original maximum rate by adding more substrate. So for the same exact reason that non-competitive inhibition, that maximum rate is decreased because it doesn't matter how much substrate we add, we're not going to reach that original Vmax. However, the Km decreases, the infinity increases because the enzyme binds to the substrate and stays bound because of the inhibitor. So that's a little bit different. The Km is actually going to decrease because, remember, these uncompetitive inhibitors can only attach to the complex. So if we have the substrate enzyme complex forming, then we finally have that inhibitor adding, it's going to be hard for that to dissociate. So that's going to increase the apparent affinity and it's going to decrease the Km. And then finally, we have mixed inhibition, which is a mix between non-competitive and uncompetitive inhibition, which means the inhibitor will bind to the substrate enzyme complex and just the bare enzyme as well. This type of inhibition, again, is not competitive. And because we are again binding to an allosteric site, Vmax is reduced in the presence of these inhibitors. So this, you should definitely know what the graph looks like. You can see that both the Km and the Vmax values are changed depending on what kind of inhibitor is used. And that's really what you should know about mixed inhibition. The other three, they'll definitely test quantitatively. Okay, so review time. Students in a biochemistry lab are running an in vitro experiment to graph the effects of a non-competitive inhibitor. Which of the following would occur after the inhibitor is added to the reaction? So pause the video, take a minute. When you're done, unpause, and we'll go over it. So for A, the Km would increase and the Vmax would decrease. That's not correct because the Km is not increasing. The Km is remaining the same. B is incorrect. The effective affinity between the substrate and enzyme would decrease. That's incorrect because effective affinity is inversely related to Km. So Km we know is staying the same. So the effective affinity is also going to stay the same because that binding hasn't been affected. So B is incorrect. C, the Km and Vmax both remain the same throughout the reaction. So the Kmax does remain the same, but Vmax decreases. The only one that's correct is D. The effective affinity between substrate and enzyme would remain the same, and the Vmax would decrease. So D is the correct answer.